Welcome back to Anatomiqa. In the previous 10 sessions, we discussed about the different aspects of bone like functions, gross structure, classification, terms used to describe bony features, parts of a young growing bone, blood supply of long bones, microstructure, fracture and healing, development and ossification, loss of ossification, and finally clinical anatomy. In this series of sessions, we will discuss about the various aspects of muscle tissue. The learning objectives of the session are: understand the parts of the muscle, describe different types of muscle fibers, describe arrangement and fascicular architecture of muscle fibers, discuss the blood and nerve supply of muscle fibers. Muscles are primarily designed for movements which are distinctive outward characteristics of animal cell. One of the fundamental properties of animal muscle cell is contractility, and this is developed in highly specialized form. Muscles can be regarded as the motors of the body. The word muscle is derived from the Latin musculus, which means a little mouse. This is probably due to the fact that certain muscles bear fancied resemblance to mice and tendons represent their tails. All muscles of the body are developed from mesoderm except the arrectoris pylorum, muscles of the iris, and myoepithelial cells of salivary, sweat, and lacrimal glands, which are derived from ectoderm. In human beings, muscles are of three varieties: unstriped or involuntary or smooth muscle, cardiac muscle, and striped or voluntary or skeletal muscle. Let us discuss the three types of muscles in detail. Unstriped muscles do not exhibit cross striations and are structurally the simplest type of contractile tissue. They respond slowly to a stimulus and are capable of sustained contraction. The unstriped muscles are also known as smooth or visceral or involuntary muscles because they are supplied by autonomic nerves and are not under the direct control of will. The smooth muscles are widely distributed in the body and maintain the normal physiological equilibrium by providing the motive power of digestion, circulation, secretion, and excretion. The smooth muscles are present in the walls of hollow-tubular and saccular viscera, ducts of exocrine glands, blood vessels, tracheobronchial tree, in the stroma of solid organs, iris, and ciliary body, the arrectoris pylorum, and sweat glands of the skin. Unstriped muscles are developed from splanchnic mesoderm except the muscles of the iris and the arrectoris pylorum of skin, which are ectodermal in origin. Now, coming to the arrangement of involuntary muscles, in hollow viscera, the smooth muscles are arranged in sheets or layers which are held together by areolar tissue. The disposition of the fibers is organized according to the functions of the viscera, and following arrangements may be observed. The blood vessels require frequent alteration of the site of the lumen to adjust the blood flow by constriction or dilatation of the vessel wall. Hence, the muscle fibers of their walls are disposed predominantly in circular manner. To propel the contents along the tubular viscera in one direction only, the muscles are differentiated mainly into two layers, longitudinal and circular. This type of downward movements along the tubes is known as peristalsis, which is observed typically in the whole length of alimentary canal, in ureters and uterine tubes. The circular muscle constricts the tube to prevent the backflow, whereas the longitudinal muscle shortens and dilates the tube distal to the constriction to accommodate the advancing contents. In saccular viscera, where movements are complicated, the muscles are distributed irregularly into three ill-defined layers. muscles of the uterus are arranged into outer longitudinal, middle circular, and inner reticular or oblique layers. Each layer is concerned with separate function. The longitudinal muscle helps evacuating the uterine contents, particularly at the termination of pregnancy. The circular muscle retains the products of conception until the full term is reached, whereas the reticular layer of muscle constricts the blood vessels and helps in homeostasis during the separation of the after births or during the bleeding phase of menstrual cycle. The unstriped muscles are supplied by both wings of autonomic nervous system, parasympathetic and sympathetic. One is meant for relaxation and the other for contraction of the muscles. Both systems contain motor and sensory components. The nerve fibers form plexuses, often intercepted by ganglion cells, and finally, as postganglionic and non-myelinated fibers reach the muscle cells. The unstriped muscles retain automatic and spontaneous contraction even when disconnected from their nerve supply. In this connection, the innervation of the intestine deserves special mention. The nerve supply of the intestine consists of two parts, extrinsic and intrinsic. The extrinsic nerve includes vagus and sympathetic fibers. The vagal fibers, after entering the intestinal wall, make synaptic junctions with the ganglion cells of Auerbach's or mesenteric and submucosal or Meissner's plexus. The sympathetic fibers are the postganglionic neurons originating in the celiac ganglion. They terminate directly on the smooth muscle cells and on the walls of blood vessels and do not enter into synaptic junction with the cell of these plexuses. The intrinsic nerve consists of Auerbach's or mesenteric plexus and Meissner's or submucosal plexus. The Auerbach's plexus is located between circular and longitudinal layers of muscles, and the Meissner's plexus lies in the submucous coat of the gut. Each plexus consists of groups of nerve cells and bundles of nerve fibers. The nerve cells act as postganglionic neurons for the vagal fibers only. The plexus of the intrinsic nerve is responsible for the spontaneous movements of the intestinal tract, even after the extrinsic nerve supply has been cut. But the movements of the intestine cease if the intrinsic plexuses are interrupted by the application of nicotine. The Auerbach's plexus regulates the direction of peristaltic waves of contraction. Some authorities claim that intrinsic plexus represent complete reflex arcs. The axons of the ganglion cells divide into two branches. One branch receives sensory stimuli from the intestinal mucosa and transmits the impulse to the other branch without passing through the cell body. The impulse thereupon reaches the muscles, the blood vessels, and the glands of the gut and produces motor activities. The extrinsic nerve supply regulates the intrinsic neuromuscular mechanism that determines the movements of the digestive tube. More recently, it is observed that the cell bodies of the neurons of Meissner's plexus act as bipolar sensory neurons. Their inner fibers convey the sense of distension from the mucous membrane, and outer fibers make synaptic relays with two sets of motor neurons in the Auerbach's or mesenteric plexus. One set consists of cholinergic fibers, which on stimulation increase peristalsis and secretomotor activities of the glands. The other set involves the peptidergic neurons, which on stimulation inhibits peristalsis and secretomotor activities by releasing ATP-like substances. Coming to the transmission of the waves of excitation, according to this, the unstriped muscles are classified into two functional groups, multi-unit type and single unit type. Multi-unit type is found in the muscle coat of the vas deferens. In this type, the smooth muscle fibers are completely separated from one another and there is a nerve ending for each muscle cell. On arrival of nerve impulse, all the muscle fibers in a given area contract simultaneously. Single unit type, also known as visceral type, is found in the intestine. The nerve impulse arriving at one muscle cell is conducted rapidly from this cell to others so that they all contract more or less simultaneously. The wave of excitation is transmitted by mechanical pull from cell to cell through the fused cell membrane or intercellular protoplasmic bridges. Now, to see the growth and regeneration, usually unstriped muscles do not proliferate by mitosis. The physiological hypertrophy of the muscles of uterus during pregnancy is due to the enlargement of individual fibers, although some authorities claim that a few cells increase in number by mitosis from undifferentiated mesenchymal cells. To discuss myoepithelial cells, they are spindle-shaped epithelial cells possessing contractile properties. We are discussing myoepithelial cells under the muscular tissue because of its contractile properties. The myoepithelial cells are developed from ectoderm and are found in the lacrimal, salivary, sweat, and mammary glands. The location of myoepithelial cells they are sandwiched between the basement membrane and the surface epithelium of the acini of glands. Electron microscope shows the presence of actin filaments within the myoepithelial cells. Numerous desmosomes connect these cells with the adjacent tissue cells. With this, we complete the location, gross features, arrangement of fibers, and nerve supply of first type of muscle, that is the unstriped muscle. In the next session, we will discuss on the gross features and nerve supply of cardiac muscle.